ever since Darwin, we've known that the early atmosphere was carbon dioxide. And unfortunately, this was lost with Urey assuming, probably in a little bit of a panic, what to do with Stanley Miller, when his new PhD student in 1950. And Urey said, well, let's assume that the early atmosphere was methane. There's never been a like Jupiter. There's never been a geologist to think that. It's always carbon dioxide. At high temperature, carbon dioxide and other oxidized species come out of volcanoes. Carbon dioxide, water, sulfur dioxide, nitric oxide, and so forth, and phosphate. So we know that. So we've got an oxidized exterior to the Earth, and yet we've got an interior that's electron-rich, if you like. It's a ferrous iron and maybe a little bit of native iron, and of course native iron in the core itself. So we've got this kind of mismatch between an oxidized exterior and a highly reduced interior. It's rather like a bacterium, actually. Indeed, geochemically, if there's no life on this planet, we know that we can generate methane from hydrogen plus carbon dioxide makes methane plus water. And it's rather like a methanogen, which does the same thing, only it does it much quicker. So we could think of life kind of bubbling off from the surface of the Earth just because of that tension, that mismatch between the alkaline solution, which has got hydrogen in it, and the acidic exterior. So if you look at this, we think of the bubbling off on the left-hand side, and that's comparable to a bacterium, which has also got slightly alkaline on the interior. It actually pumps protons outside to the exterior, and the protons come back. It's called the proton motive force. All life uses the proton motive force. All life uses it to make ATP, or at least pyrophosphate, and that's what energizes us. We probably make nearly our body weight of ATP every day. And that's made through the protons coming through the membrane. So it seems to me that we would need that in the initial condition. We can't invent something like the proton motive force. It has to be there. And there's other things like the redox differences between very reduced or hydrogen-rich interior. We have a hydrogen-rich interior. We use NADPH, for example, to generate hydrogen, to give hydrogen to us for our metabolism. This is very similar to the little bubble I've got on the left here, the kind of entropy trap where the fluids, the two different fluids interact. So the demon that does this work is something called serpentinite, or it's a serpentinization reaction. It's the only thing you need to know about geology in this talk, and that is that the fluid comes down into the interior, and the lavas are changed to something called serpentinite, and that's that kind of ultimate ancestor, if you like. And just to emphasize what a common rock this is, this is the back of the United Nations building, and that's serpentinite. You'll see it on many shop doorways. And the reaction here shows that carbon dioxide can be reduced slowly but continually to methane, actually geochemically. And what I'm emphasizing is that biochemistry is not just better geochemistry. We've got to try to work out how we get a transition from doing this kind of job from CO2 to CH4, which is what the world wants, and how life comes in and tries to quicken that reaction. Basically, both magnesium ion silicates and water interact to make the hydrogen and the hydroxyl, and also some methane. But there's also something else that can be made, and that is acetic acid or vinegar. That's the kind of halfway mark, and I think that's pretty significant, that point. So when black smokers were first discovered, understandably, the people who discovered them, Jack Corliss and John Edmund, thought maybe life started here, because here's the black smoker. I don't know if you can see them, but there's crabs there, there's all kinds of worms. There are oases at the bottom of the ocean at these very hot springs. Now, 400 degrees centigrade, and obviously too hot for life to have started, although life, once it has started, can populate the exteriors. But here it's interesting, you can get all those metals that you need, the iron, the zinc, the nickel, the molybdenum, the tungsten, all coming out of this spring. And they're called black smokers because the water, the metals in there start to precipitate as they come out and make this black smoke. Actually, if you go and take that water and take them onto the deck of the ship, nothing happens until you actually add the seawater. So it's a precipitation reaction brought about by a pH change. Otherwise it remains supersaturated. So in other words, the early ocean, because the early ocean was acidic, you wouldn't have had this black smoke. You would have had a supersaturation of all these metals that are required by enzymes. So those were the acidic hot springs, 400 degrees centigrade, you see in the center there. But we, working as geologists, had found some other kind of springs which were much cooler, and we predicted that there should be some off-axis springs away from this. And in 1999, Lost City was discovered, and that satisfies nearly every aspect of our expectations. The hydrogen's up to 15 millimoles, there's methane up to 2 millimoles. They last for 30,000 years, that's 10 to the 17 microseconds. To my mind, plenty of time for life to get started. The only thing missing there is sulfide. So, the reason why there's life is because it's uphill to go from CO2 through formate to formaldehyde. That's the uphill part in terms of kilojoules. So it's endergonic reactions. And what we need, of course, is to have exergonic reactions. So once you get to formaldehyde, then it's all downhill to methane, or halfway to the methyl group, like as in methyl alcohol. But you've got to get up to that top there. And of course, thermodynamically, a little bit of formaldehyde is made in the deep crust, and therefore you do get a kind of geochemical siphon, bringing this, making this reaction go to methane, but it goes very slowly. But what we think is one way through this, it's extraordinary that life requires molybdenum and tungsten. I mean, you know, molybdenum is element 42 and tungsten 74. I mean, this is way beyond iron to make these, but we need these. And why do we need them? Is because we need a two-electron system, a bifurcation, so that we can offer the inside of this bubble electron acceptors on the outside. And so carbon dioxide doesn't really want to have another electron in it, and formate doesn't like to go to formaldehyde. It's energetically very uphill. But if you have something like nitric oxide or ferric iron on the outside as an electron acceptor, then because molybdenum likes to be either 4 plus or 6 plus, it loses one electron to nitric oxide, and then immediately the other one can be lost to the carbon dioxide. And that's the reason why we think molybdenum and tungsten are required, molybdenum or tungsten are required always in the simplest pathway of life. The simplest pathway of life is called the acetyl coenzymase synthase pathway. It's the cheapest, and it's a downhill single pathway. It's not a cycle. We don't need hypercycles because we've got a low entropy system to start with. So we can take this to the lab very quickly. We can reproduce the conditions on the early Earth. We can inject the fluids that we make in a hydrothermal system into what we call a Hadean ocean. That's 4.4 billion years ago, and immediately we get this hydrothermal mound. And this hydrothermal, at this system, we can make methane just like the Earth does through serpentinization reactions, and we can generate sulfide. So that's the important point. And generating sulfide means that we can generate those kind of enzyme-like minerals that are extraordinarily similar to the centers of the active sites of enzymes. And these are the kind of structures we make. Those, if we take the kind of little black mound you see there on our putative sea floor, and on the right, you can't see them very well, but you can see these quite nice little compartments made of iron, nickel sulfides, and silicate. So these are this is the first membrane. These membranes are inorganic membranes. There's no organic molecules on the early Earth, so we don't have organic molecules to play with. In spite of people wanting lipids. I mean, lipids aren't much use. They're like a plastic bag anyway. The whole thing about the way membranes work is it works through proteins. So it's the proteins that matter. What we need, first of all, is proteins, or perhaps their precursors, peptides. But up until then, first of all, we have this inorganic membrane, and these are the kind of culture chambers of life. So the whole model just rests actually on this particular diagram. On the bottom, we've got the hydrogen, the methane, and hydrogen sulfide coming up, maximum 120 degrees centigrade, pH around about 11, very alkaline. It comes in and interacts with the carbonic fluid, which is just like carbonated water, pH 5, 5.5, they interact and immediately you get this precipitation, which we can do in the lab and which we can see in the ocean. At the interaction, we get these kind of membranes on the margins, and at the margins, we've got these proton gradient, which is about 5 units, which equals about 300 millivolts. But also on the outside, you've got things like nitric oxide or nitrate. You've got phosphate in the ocean. So we've got places for the electrons within the hydrogen to go to on the way to reducing the carbon dioxide to, in the first case, we would say acetic acid or acetate. And the whole thing happens in these little compartments. So this, to us, these are the first reactions of life. They're very obvious geochemical reactions, and they're the same reactions as the simplest pathways of life. So what do these inorganic structures look like? They have some real beauty that's suggesting these kind of 4-micron waves on their margins. And we presume that this is telling us something about what's happening chemically, although we're not sure what is happening chemically. On the inside, you get these structures called framboids, which is like the French raspberries. Those are kind of far from equilibrium precipitates. On the outside, you tend to get dendrites, and within the membrane, you actually get little compartments even within the membrane. And we think that right in the membrane, that's where the first business, so to speak, is done. In fact, even in life today, much of the actual metabolism goes on at the membrane. We weren't going to set out to work on the origin of life. It didn't occur to us. But finding these small chimneys in Ireland, 350 million years old, that's what started us off. And I realized, of course, I was going to have to start learning biochemistry, biology, evolution, more chemistry, more physics. I never realized I was going to have to learn something about quantum mechanics. But apparently, and a paper has just come out to this effect, using our hypothesis. I was trying to bone up on it last night, but it was just hopeless. I mean, so I promise you, when I leave here, not only am I going to be listening to you all about quantum entanglement and decoherence, I'm going to get down to it. But apparently this is very helpful in terms of the search for catalytic effect within the membrane. And this paper by Hayden, McGlynn, Robus, Origins of Life, just come out. I happen to have it on my memory stick, so if anybody's interested, please take it from me. So I'm going to skip this one because I really, and I'll come back to it another time perhaps. But so here's the kind of minerals we make easily in the membrane. This is something called gregite. And all you have to see in that bottom right-hand corner, there's something that looks like a cube. Isn't that fair? So there's four ions and 4 sulfurs make up this lovely cube. Green green is the iron, and yellow or orange is the sulfur. And you go out through 2 sulfurs upwards to either nickel or iron, another green one. I've seen that there. And there's our membrane on the bottom left-hand side. Now we can make this within that membrane. But just out of interest, look at this. This is the acetyl coenzyme synthetase, and also the carbon monoxide dehydrogenase. They're the 2 things that really matter. You've got to be able to get carbon dioxide to carbon monoxide, and then you've got to be able to take the acetate, the methyl group, and add it to the carbon monoxide, and they've both got a very similar structure. And look at the similarity between, there's that cubane structure on the bottom, on the right-hand side, and it goes off through a sulfur, that's a cysteine sulfur, up to nickel, and then there's another nickel. That other nickel probably just kind of stabilizes the system, and everything happens on that nickel. So here we've got a structure that is just like life needs. So all we would need to do is to kind of encapsulate that in some kind of peptide. And here we can say, this is something called ferredoxin, which is fer, as in iron, redox in. So it can switch between 2+ and 3+. Now I should have pointed out, can I go back to that? So the point about this structure here is that, to put it crudely, the ions in the box there, in that cubane structure on the left, they're about Fe 2.5+, so to speak. The electrons are delocalized, but it's between 2 and 3, and it's a forgiving structure. If you take out that electron, all that happens is that it starts to contract a little, and if you add an electron, it expands a little. That's the only, but it doesn't change topology. And that's, of course, that's how that enzyme works. Now, then we can show, if we can get a small peptide, and we can make peptides in that hydrothermal system you've just seen, we can make amino acids, we can make short peptides. The peptides can then, on the backbone, it doesn't have to be the side chains, the backbone, then through hydrogen bonding can go to the sulfurs on iron, and you can get something that looks like a ferredoxin, which has probably got the deepest of all pedigrees of the proteins, and you can make that without any coding. Even more significant is the phosphate. Now, the phosphate is what I've just been talking about, the ATP. We need adenosine triphosphate. Now, this is pyrophosphate. The adenine doesn't really matter that much. What matters is having the phosphate as the energy source. So if we can have a phosphate and then we can protect it through the backbone of a short peptide, in this case a 5-mer peptide, then we've got something that looks exactly like, really exactly like ATPase or pyrophosphatase in all bacteria and all archaea. So we didn't have to do anything, we didn't have to do any coding. It's just that these two different structures recognize each other, and we get this kind of pickup, this kind of ligation of the phosphate, and then we protect that phosphate solution, and we can also it can encourage making pyrophosphate. Now, that's quite a complicated process, but basically the way it works is that this is all side chain. All you're looking at here is a phosphate or two phosphates. Of course, they're repelling each other because they're both negative, so you have magnesium in the solution to come up, and that helps to the two negatives so that the phosphates can interact with each other to make pyrophosphate. And the way that happens is through using a proton. So on the lower diagram you can see magnesium phosphate plus the proton makes the diphosphate, or magnesium diphosphate. And that's what can be used to make more polymer. So we kind of got a feedback system of, you can make polymers, a peptide. This helps to ligate the phosphates, protect it, and allow it to become pyrophosphate. And then the pyrophosphate can make more peptide. So we've got this positive feedback effect. Now I haven't talked about, obviously, the code. Now, and of course it's much too complicated to get into that, but I can just say one thing about it, and that is that I talked about adenosine triphosphate. Now, and I've also talked about nicotine-amide diphosphate. Now they're phosphates, but they're nucleotide phosphates. They are like, for example, ATP is like, or the monophosphate is just like part of an RNA. So it seems to me that the way you get into the RNA world is to basically every molecule that is generated in the system has to have a job to do it. If it doesn't have a job to do, it's blown out with the waste, out into the ocean. But if it has a job to do, then eventually these nucleotides, these phosphate nucleotides can join up to make RNA. So eventually you can segue into an RNA world without having to think, how do we get a code? The parts of the RNA that had jobs to do before they joined up to make RNA. So the way we would see then the whole thing is that we've got a hydrothermal system at the bottom of the ocean. We make these compartments. The compartments allow the interaction between the two fluids, the hydrogen and the carbon dioxide. It brings the molybdenum, the tungsten, and the nickel and the iron that are all important. It makes these small compartments, and this builds up into a synergy between things like the peptides we make and the inorganic elements that are present. We go through an RNA world, perhaps eventually to DNA, and immediately we get a split between, and what is remarkable, we have a bifurcation right at the very beginning of life between the archaebacteria and the true bacteria, and they really are different, completely different. There's three domains of life: the archaebacteria, the true bacteria, and the eukaryotes such as us and trees and grass. So we've got this bifurcation right at the beginning, and we would suggest the bifurcation is due to the geochemistry. The geochemistry, you can either go to acetic acid, we've seen how to do that, or you can go to methane. Or indeed, you can make what we think actually, you can go, the first thing to do is to burn the methane. So on the right-hand side, we say the archaea, reacting with nitric oxide, for example, can make some carbon dioxide and water and nitrogen, and that's a very slow process, and it makes these sturdy organisms called the archaebacteria. And on the left-hand side, the more versatile bacteria, and they started off as acetogens. So those are the two aspects of this. Now, this is it. Yes. So I've been in LA about 5 years, and I have to say I love it. And people ricochet off each other in LA, and you might go to a party and so forth. And eventually, of course, probably I will start talking, and I'll say, what do you do? And I say, well, I work on the origin of life. And that may or may not start the conversation. But 6 months later, if I meet them, they'll say, and this has happened several times, oh, you're the guy who's working on the Big Bang. And I said, well, actually, no, I'm not. But of course, it did put me in mind of what Sean works on, and many of you, and there are similarities. I can see why people think of it. We have one advantage. We know what's going on before the Big Bang. So we've got a nice low entropy system, hydrothermal system, which we can trust, and it's aqueous geochemistry. But as I said again, biochemistry isn't just better geochemistry. It is really different. So what causes a transition to this new phase, to metabolism? And the transition, we think, is through the proton-motive force, which gives us 300 millivolts, which makes our pyrophosphate, and also electron bifurcation through molybdenum or tungsten to reduce nitric oxide, for example, and carbon dioxide. By the way, if there were no carbon dioxide, it would just reduce nitric oxide, of course, and that would be the end of it, and there would be no life anyway. But because there's so much carbon dioxide, then, of course, it's very easy for the second electron to go to what's available, and that's carbon dioxide. So that's the way into metabolism, presumably partly through quantum entanglement and decoherence as well, but you'll forgive me if I just leave that to one side, but I do think that's perhaps 3 legs of the stool that takes us through to metabolism. And then from metabolism, we need to get through to biochemistry proper, and biochemistry proper only starts when you've got an RNA coding or eventually DNA, and that's done through coenzymes, basically. That transition is through coenzymes joining up together to make an RNA world, and the RNAs can interact with peptide or protein, or sorry, I should say with the amino acids to start to join them up to make a coded peptide. So we started off with an uncoded peptide, and now we go to a coded peptide when we get into biochemistry, and that coded peptide, of course, is a protein. And they're small, and they get bigger and bigger for all kinds of reasons in time. Thank you.